The cell is the fundamental unit of life. Most living things on Earth are single-celled organisms. We begin life as a single cell, the fertilized egg, but today we are multicellular organisms composed of trillions of cells, all of whom originated from the fertilized egg. Cells were discovered in the 17th century following the invention of the microscope. But the significance of the cell was not appreciated until the 19th century, when the cell theory was developed. The cell theory states that the cell is the fundamental living unit, that all living things are composed of cells, and that cells are the offspring of pre-existing cells. Cells are too small to be seen with the unaided eye, and the size of cells varies enormously. Here you are viewing the two major types of cells, eukaryote and prokaryote. Eukaryotic cells are the kind that compose our bodies. These cells are named for the presence of a nucleus inside the cell. Eukaryote means beautiful nucleus. Prokaryote cells are much smaller than eukaryotic cells. These images give you a feel of the relative size difference between them. Let's enlarge the prokaryotic cell to see it more clearly. Unlike the eukaryotic cell, the prokaryotic cell does not have an internal nucleus. In fact, prokaryote means before the nucleus. In general, the prokaryotic cell is much simpler than the eukaryote, but they are more prolific. There are more prokaryotes than eukaryotes on Earth. In fact, prokaryotes on the surface of our body and in our gut outnumber our eukaryote cells by a factor of 10 to 1. The largest group of prokaryotes are the bacteria, some of whom produce both harmful and beneficial products for humans. Here you can see a human white blood cell in pursuit of two bacteria. This image is from a scanning electron microscope that has been color enhanced. The white blood cell is creeping toward the bacteria and will attempt to engulf and destroy them. We begin our study of the eukaryotic cells of our bodies by reviewing our chemical composition. Recall that we are mostly water, about 60% by weight. A standard reference 154-pound male, or 70-kilogram male, carries around more than 90 pounds or 40 kilograms of water. Why then don't we hear the water sloshing around in our body when we move around, like water in a hot water bottle? We don't, because most of the water in our body is sequestered inside little compartments we call cells. About 2/3 of our body water, or 40% of our body weight, is inside our cells and is called intracellular fluid. The remaining third, or 20% of our body weight, is outside our cells and is called the extracellular fluid. We are more like fish than you might suspect. We do not live in water, but our cells do. This extracellular fluid that bathes our cells is essential for the survival of our cells. Cells that lose this watery environment soon die. In fact, the outermost layers of cells of our skin are dead cells because they have been pushed away from their watery environment. But even in death, they perform a valuable function, sealing water inside the body and keeping germs from gaining entry. Cells are membrane-bound, membrane-partitioned structures. The membrane that separates the inside from the outside of the cell is called the plasma membrane. However, The membranes inside the cell are far more extensive than the plasma membrane. These internal membranes compartmentalize functions inside the cell, so the cell can carry out many diverse functions. Think of a campus building. The external wall keeps the weather and the bugs out. The internal walls make it possible to teach physiology in one area, microbiology in another, and anatomy in yet another area without interfering with each other. The membrane-bound compartments inside the cell form organelles, such as mitochondria and lysosomes. The membranes of the cell have a common structure built on a molecular foundation of phospholipids. Here you are viewing a representation of a compound known as triacylglycerol, or triglyceride. This is the kind of molecule that composes fats and oils, such as butter and corn oil. The triacylglycerol is composed of a 3-carbon glycerol backbone. Each carbon in glycerol is bonded to a fatty acid, which is a hydrocarbon chain that includes a carboxylic acid. The triacylglycerol molecule is hydrophobic, meaning it is not soluble in water. You put corn oil and water together, and they will separate. The oil will go to the top, because oil is less dense than water. Fat floats. This is why it's so difficult to drown a fat person. Water doesn't dissolve fat because fat molecules are not electrically polarized or sticky. Recall, water is made of oxygen bonded to 2 hydrogens. The oxygen is so much larger than the hydrogens that the shared electrons spend most of their time around the oxygen nucleus. This gives oxygen a slight negative charge and the hydrogens a slight positive charge. We say that the water molecule is electrically polarized. Opposite charges attract, so one water molecule will attract and bond weakly to another water molecule. Thanks to this hydrogen bonding, we are a liquid instead of a gas at body temperature. Because fat molecules are not electrically charged or polarized, water has no attraction for them, so they separate into different phases. It is as if you invited people to your party, some of whom spoke English but no Spanish, and others who spoke Spanish but no English. You would end up with 2 parties based on the language differences. Something similar happens when you try to mix oil and water. Because fat molecules don't dissolve in water, they are not stable in the water compartments of our body and cannot be used in the membranes of our cells. But if we replaced one of the fatty acids with an electrically charged or polarized group, we could produce a molecule that is stable in body water. Our bodies use phosphates for this purpose. Phosphate is phosphorus bonded to oxygens and have a net negative charge. Often accompanying the phosphate group is a nitrogen-containing group like choline or an alcohol like inositol. The result is a compound that is amphipathic, meaning it has a split personality, with the fatty region hydrophobic and the phosphate region hydrophilic. We draw the phospholipid like this. The blue circle represents the water-soluble phosphate end, and the 2 squiggly lines represent the 2 remaining fatty acids, which are hydrophobic, not water-soluble. If you placed phospholipid molecules in water, they will spontaneously form a bilayer. Two layers of phospholipids with the fatty acids in the center, shown here in yellow, and the phosphates facing the water matrix, shown here in blue. The result is a stable boundary that can form compartments. All the membranes of the cell have the phospholipid bilayer at their foundation. Here you see the phospholipid bilayer of the plasma membrane, separating the fluid inside the cell from the extracellular fluid. The phospholipid bilayer serves mostly as a partition. Dispersed throughout the bilayer are other compounds, most notably proteins. In this image, the phospholipids are shown in blue and yellow, and the proteins are shown in green. Some proteins may be peripheral to the membrane. Other proteins may be integral, in other words, span the entire width of the membrane. The membrane proteins give the membrane its dynamic characteristics. This model of the membrane originated in the 1970s and is called the fluid mosaic model. Fluid implies flexible as opposed to rigid. Mosaic implies a variety of compounds are present. Here are some of the major functions of proteins in membranes. Some proteins serve as enzymes, or the agents of chemical change. Many cells have in their plasma membrane an enzyme called cyclooxygenase. This enzyme converts fatty acids into chemical messengers. Among its many functions, cyclooxygenase helps clot the blood and activate pain receptors. When you take a non-steroidal anti-inflammatory painkiller like aspirin, it binds to cyclooxygenase, preventing it from producing compounds that activate pain receptors. The result is your pain goes away. But aspirin also inhibits cyclooxygenase from producing chemical messengers that help your blood clot. That is why you are advised not to use aspirin several days before a planned surgery. Your doctor doesn't want you bleeding all over their clean sheets. Other proteins act as receptors or antennae for the cell. For example, in the membranes of photoreceptor cells of the retina of our eyes, we have a group of proteins called opsins that respond to light by generating signals. The hormone insulin works by binding to receptors in the plasma membrane of its target cells. Because the phospholipid bilayer is largely impermeable to many compounds, proteins may act as pores or channels to allow substances through. In some cases, the proteins bind a solute on one side of a membrane and escort it through the membrane. Our focus initially will be the membrane that separates the inside from the outside of the cell, the plasma membrane. Please don't confuse the plasma membrane with the cell wall. Human cells do not have cell walls. Cell walls are rigid structures found in plant cells and bacterial cells. Animal cells are surrounded only by a flexible plasma membrane. One of the critical functions of the plasma membrane is to regulate what enters and leaves the cells. If the plasma membrane ruptures and the extracellular and intracellular fluids freely mix, the cell will die. The plasma membrane maintains a difference in composition between the extracellular and intracellular fluids. This difference in composition is essential for cell survival. Let's compare the composition of these two fluid compartments. Outside the cell, we have more sodium ions than we have inside the cell. The symbol for sodium is capital N lowercase a. This is from the Latin word for sodium, which is natrium. We can't use the letter S because S has been used for another element, sulfur. Note that sodium has a single positive charge. It's a monovalent cation. Chloride is more concentrated outside the cell than inside the cell. Chloride has a single negative charge, a monovalent anion. Glucose is more concentrated outside the cell than inside the cell. And calcium ions are more concentrated in the extracellular fluid. Calcium is a divalent cation, two positive charges. Inside the cell, we have more potassium ions than outside the cell. The symbol for potassium is capital K. The K is from the Latin name for potassium, which is kalium. We can't use the letter P to symbolize potassium because that's been used by another element, phosphorus. Proteins are more concentrated inside the cell than outside the cell. The charge on proteins can vary depending upon the amino acid composition of the protein and the environment of the protein. Phosphate is more concentrated inside the cell and has a net negative charge. Magnesium ions are more concentrated inside the cell. Magnesium is a divalent cation, two positive charges. Sulfates, sulfur bonded to oxygen, are more common inside the cell than outside the cell. Sulfates are also negatively charged. This difference in composition between the extracellular and intracellular fluids is necessary for the survival of the cell. If the plasma membrane should rupture and the extracellular and intracellular fluids freely intermix, the cell will die. The extracellular and intracellular fluids of our body are complex solutions. Solutions are composed of two phases. The dominant phase is called the solvent. In life, the universal solvent is water. Dissolved or suspended in the water matrix are solutes, like glucose or calcium ions. Most solutes cannot cross the plasma membrane without the assistance of proteins. Water molecules can cross by squeezing between the phospholipid molecules of the membrane, or with the help of proteins that form aquapores, or water channels. This image illustrates the relative permeability of the phospholipid bilayer to different solutes. Note that small, nonpolar solutes, like molecular oxygen and carbon dioxide, can readily cross the bilayer without assistance. Small polar compounds, like water, can squeeze slowly through the bilayer. Large polar compounds, like glucose and sucrose, cannot cross the bilayer without assistance. This same impermeability applies to ions like sodium and potassium. To enable these charged and polar compounds to cross the membrane, we rely on the proteins within the membrane to act as channels or carriers. There are four major means whereby substances can cross the plasma membrane. These include simple diffusion, facilitated diffusion, active transport, and bulk transport. We'll look at each of these in turn, beginning with simple diffusion. Diffusion is the process of spontaneous movement of substances from regions of high to regions of low concentration. To understand how diffusion occurs, you need to understand that atoms and molecules are in constant motion. When we take the temperature of a solution, we are measuring the rate of motion of the molecules in the solution. The warmer the temperature, the greater the rate of motion, or kinetic energy. The molecules in a solution are continuously bumping into each other and tend to spread out to fill the available space. If we enlarge the available space, the molecules would spread out to fill this additional space. This is what we call diffusion. Diffusion occurs if there is a concentration gradient and a path for the molecules to spread out. If you brought home from the store a solid air freshener and opened it, you create a pathway for diffusion to occur. First, your room smells nice, then your whole house smells nice, and in a couple of days, the neighbors call to ask if someone died. That's diffusion. After a few weeks, you don't notice the smell anymore, so you check on your solid air freshener and find that only a stick remains. All the smelly molecules have diffused away. You could get your butterfly net to try to chase them down, but it would be easier to go to the store and buy a new air freshener. Even molecules in the solid state will diffuse if they are not tightly bonded. Have you ever gone to the freezer for some ice cubes, only to find the ice tray empty? You start the blame game, accusing others of not refilling the tray. But in reality, if you don't use the ice in the tray after several months, the water molecules will diffuse away. Where do they go? Many of them are clinging to the walls of your freezer. The diffusion of your ice cubes, or air freshener, is what we call simple diffusion. Because of the relative impermeability of the phospholipid bilayer of the plasma membrane, many molecules need assistance to cross. This assistance is usually facilitated by proteins, and so is called facilitated diffusion. Here you see a protein pore, or channel, in a membrane. When the channel is open, the molecules can diffuse down their concentration gradient. Why does diffusion only occur downhill, you ask? It has nothing to do with what the molecules want. It has to do with what is more probable. If there are more solutes on one side of a membrane than on the other, the probability is greater they will strike the membrane from the side where they are more concentrated. Occasionally, a solute may pass from the other direction, but the net movement is from high to low concentration. That is what we call diffusion. Many of the protein channels that permit diffusion are dedicated to one or a few solutes. For example, there are separate channels for sodium ions and for potassium ions. Moreover, these channels are gated so that diffusion across the membrane can be regulated. If the gates are open, diffusion can occur. If closed, come back another day. In some cases of facilitated diffusion, the membrane protein must bind the solute in order to facilitate its crossing the membrane. The carrier can transport the solute in either direction, So what determines the net movement, or diffusion, is the direction of the concentration gradient. Glucose is transported across membranes via this carrier-mediated transport. Sometimes we need solutes to go where they will not go spontaneously. Think of your car. You don't have to burn gas to get your car to roll downhill. That is a spontaneous process. But if you want your car to go uphill, you'll have to input energy. The same is true when cells need to move a solute from where they are less concentrated to where they are more concentrated. This requires a carrier protein, like you see here in blue, and a form of energy, usually ATP, because movement against a concentration gradient is not a spontaneous process. This is called active transport. Nearly a quarter of all the energy expended by our bodies each day is actively transporting solutes against a concentration gradient. Cells also have the capacity to move droplets of fluid or large particles or solutes en masse across membranes. This is called bulk transport. Bulk transport also requires cellular energy. There are two forms of bulk transport. The form shown here is exocytosis. In the top diagram, we have a cell that contains vesicles full of chemical messengers that are being released en masse onto the neighboring cell. This is how nerve cells usually communicate. Below, we have a cell that is releasing a membrane-bound particle to the extracellular environment. Note the vesicle membrane fuses with the plasma membrane as the particle is released. Bulk transport can work in the opposite direction. It's called endocytosis. This is where a particle or droplet of fluid is brought into the cell surrounded by plasma membrane. Some cells in our body use endocytosis to clean up debris and foreign invaders from the extracellular space. Here you are viewing two containers that contain solutions that are separated by a membrane. The blue background is the solvent, water, and the red dots are the solutes. If the membrane is permeable to the two solutions, would diffusion occur? No, it wouldn't. Why not? Because there is no concentration gradient. Note that there are 10 solutes in the same volume of solution on each side of the membrane. Would molecules cross the membrane? Sure they would. But the rate they would cross, going from A to B, would be the same as the rate from B to A. So no net transport would occur. Diffusion refers to net transport. Let's change the scenario by adding more solute to B. If the membrane were permeable to solute, would diffusion occur? Yes, it would. Solute would move from B to A until equally distributed in the two containers. But what would happen if the membrane were permeable only to the solvent, water, and not the solute? Let's check it out. Note what happened. The solute couldn't move down its concentration gradient, but water could. So water diffused from A to B. We call the diffusion of water across a semi-permeable membrane osmosis. Osmosis is a type of diffusion. When we added more solute to B, we created two concentration gradients. One was a solute concentration gradient directed from B to A. The other was a solvent concentration gradient directed from A to B. The membrane prevented the solutes from diffusing, but not the solvent. Think of an elevator. The more people you cram into an elevator, the less space available for the solvent, air. In a crowded elevator, you may have to wait for someone to exhale so you can inhale. When we added more solute to B, we raised its concentration, but we lowered the solvent concentration in B, creating a gradient that resulted in water diffusing from A to B. Osmosis. Osmosis is important to cells because the plasma membrane is normally permeable to water, but usually impermeable to most solutes. If there is a change in the non-penetrating solute concentration in the extracellular fluid, the result is water will move into or out of our cells. If water leaves the cell, it may shrivel up and die. If water enters the cell, it may bloat and explode. That is why we have a homeostatic mechanism for regulating the osmolarity of the extracellular fluid. Osmolarity refers to the concentration of non-penetrating solutes in a solution, or, inversely, the water concentration of a solution. Here you see what can happen to cells if placed in extreme osmotic environments. Here we have a red blood cell in its normal environment. Red blood cells are rather odd. They have a thinner center than edge. We call them a biconcave disc. If we take a red blood cell out of its normal environment and place it in a concentrated salt solution, a so-called hypertonic solution, the salt can't diffuse into the cell through the plasma membrane. So what happens is water diffuses out of the cell and the cell shrivels. That's called crenation. Now if we take our red blood cell and drop it into a pure water solution, a distilled water solution, a solution which has no solutes at all, just water. The solutes of the cell can't diffuse out of the cell down their concentration gradient. The plasma membrane prevents that. So what happens is the water diffuses into the cell swells, and it may burst. We call this kind of a medium that causes water to diffuse into the cell a hypotonic medium. Here's some animation showing you what happens when we place blood cells in different environments. First, we're placing our red blood cell in a hypertonic solution, like a concentrated salt solution. The salt can't get in, so the water diffuses out. Here we're placing it into a hypotonic solution, like pure water. Since the solutes in the cell can't diffuse out, the water diffuses in, and the cell here bursts. And here we're placing the cell in an isotonic solution. An isotonic solution does not cause the shifting of water into or out of the cell. Isotonic, meaning the same tonicity, the same osmolarity as you would find in the normal extracellular fluids. So here's a question for you. What happens if we place a blood cell in a hypotonic solution? So the red circle is our red blood cell. If it's a hypotonic solution, what does that imply about it? It implies that it has a high water concentration in the environment and a low solute concentration. So most of the solutes, the higher solute concentration, is inside the cell. The higher water concentration is outside the cell. So what's going to happen? Well, since the solutes in the cell can't diffuse out, what happens is the water outside the cell diffuses down its concentration gradient into the cell, causing the cell to swell and perhaps burst. Well, it's not good to have your cells swelling and bursting or shriveling up. And so we have homeostatic mechanisms for regulating the osmolarity, the water concentration, of the extracellular fluid. Let's say your bar has recently opened, and so you're going to celebrate, and on the bar there are free salty snacks. Your bartender encourages you to have more, eat all you want. So the bartender is loading you up with salt. Salt, sodium chloride, is a substance that's readily absorbed from the GI tract. Nearly all of it is absorbed from the GI tract into the bloodstream, forcing the kidneys to try to get rid of it. On its way to the kidneys, however, this rising tide of salt in the body causes water to diffuse out of cells. Our cells start to shrink as a result of consuming all this free salty snack. Fortunately, we have receptors in the hypothalamus of our brain, the so-called osmoreceptors. As they shrink due to water leaving the cells, they send out an alarm. One of the effects of that alarm is the release of a hormone called antidiuretic hormone that acts on the kidneys to tell the kidneys to conserve water. But the kidneys only conserve water that's already in the body. Another effect of the osmoreceptors is to drive you to drink. They activate your desire for fluid, your thirst. And so what do? You say, "Bartender, I would like a glass of water." Then the bartender laughs at you. "We don't serve water here, but we'll sell you a beer for $5. That was the whole point of feeding you the free salty snacks, to load you up on salt, causing water to diffuse out of your cells, in particular the osmoreceptors in your brain, so you'll get thirsty and have to order an expensive drink. Your bartender is manipulating the osmolarity of your extracellular fluids for personal financial gain. Shame on them. Viva la revolution. Suppose you were dehydrated and needed intravenous rehydration. Or suppose you needed medication that had to be administered intravenously through a significant volume of fluid. You would not want this fluid into your bloodstream if it's going to cause cells to swell or cells to shrink. Consequently, they make standardized solutions that when injected into the bloodstream will not cause any significant shifting of water into or out of the cells. For example, if you take 900 milligrams of sodium chloride in 100 milliliters of solution, you get a 0.9% sodium chloride solution, a so-called standard saline solution. If you inject this solution into the bloodstream, it does not cause significant shifting of water into or out of the cells. You can make a similar solution out of glucose. You take 5 grams of glucose in 100 milliliters of solution, producing a 5% glucose or standardized glucose solution that also will not cause any significant shifting of fluid into or out of your cells. There are two modifications of the plasma membrane that some cells express. One of these you're looking at here is called microvilli. Microvilli are folds in the plasma membrane that are found primarily along the small intestine. These folds dramatically increase the surface area of the small intestine. That's important because most of the nutrients that we absorb from our food are absorbed through the lining of the small intestine. If these microvilli were to disappear, you could eat normally but starve to death. Another modification of the plasma membrane in certain cells is cilia. Cilia are more like hair projections, tiny hairs that project from the surface of cells. We have cilia, for example, projecting from cells along the respiratory tract. The function of these cilia is to sweep debris away from the lungs. Females have ciliated cells along their uterine tubes. The function of these cilia is to sweep the egg along towards the uterus, because the egg has no means of mobility of its own. The sperm actually has to swim against the sweeping action of these cilia. In our next video lecture, we'll be taking a tour of the organelles inside the cell.